Good evening, everyone. A very warm welcome to all the respected dignitaries, faculty members, researchers, professionals, scholars, and dear participants join us from different parts of India and abroad. On behalf of Edu-Mentors Pvt Limited, it is my pleasure to welcome you all to the 21-day International Summer School Training Program on Climate Resilient Agriculture and Allied Sciences. It is my privilege to introduce the visionary behind this initiative. Dr. Indu Sharma is an associate professor in the Department of Life Sciences at the University Institute and Sciences and Dean Alumni and Extension Activities at Sant Baba Bhag Singh University, Jalandhar, Punjab, India. Dr. Indu Sharma, gold medalist and PhD from the Department of Botanical and Environmental Sciences, Guru Nanak Dev University, Amritsar, Punjab, India. Dr. Sharma is the recipient of DST JRF Fellowship and DST Inspire Fellowship from DST Ministry of Science and Technology, Government of India. She has 14 plus years of teaching and research experience in the field of plant stress, psychology, biochemistry, and molecular biology. Dr. Indu Sharma is published more than 70 research papers, review article, book, chapter, and journal books of international repute. She is currently guiding 6 PhD scholars, notably Dr. Sharma published a research paper as the first author in the journal with an impressive impact factor of 8.8 2013, which is indexed in the Science Citation Index. She has edited 2 books indexed in Scopes and is currently working on 3 more with internationally recognized publisher. Dr. Indu Sharma is also inventor of 5 national patents. Additionally, Dr. Sharma has compiled the data and edited both volumes of SBBSU newsletter. Beside these, Dr. Indu Sharma, external examiner and paper setter of various exams conducted by GNDU and Khalsa College, Amritsar. We are truly honored to have such an accomplished academician with us today. I am confident and that today's session will be highly informative, inspiring, and beneficial for all participants. Without taking much more your valuable time, I respectfully invite Dr. Indu Sharma to deliver today's lecture. Ma'am, the virtual platform is all yours. Welcome and thank you for joining us.
Thank you, ma'am. I really appreciate your efforts that you people are organizing online lectures and combining a number of researchers, scientists, as well as academicians to share their knowledge with the youngsters. So if I'm audible to you, may I proceed further?
Yes, ma'am.
I've already shared my
profit to payment of.
Anji?
Hello.
Hello.
Confirm and send billing details. Hello. ये भर देना, ये हम अपना ही रहने दे, कोई दिक्कत नहीं होगा, हमारे नाम पे रुक पाएंगे वो तो.
I think somebody needs to be muted.
Dear participants, please turn off your mics.
Okay, please check whether my presentation has been shared with all of you. Madam, is it visible?
Okay. Ma'am, slideshow on mode kar do.
Okay, so is it visible to all of you?
Yes, ma'am.
Okay, so today's topic is PGPRs for sustainable agriculture. Why I have chosen this topic? Because agriculture is essence of life. And nowadays, we people go to market, we target that we should eat the fresh vegetables, we should have the fresh raw fruits. And everybody, when they go to market, they procure really very good quality of the fruits and the vegetables. in order to improve our health, in order to prevent the diseases, to enhance the nutrition in the body, or to increase their immunity. But what happens that we do not know whether these vegetables or the fruits which we are getting from the market, are they healthy for us or not? Why? Because in the fruits, in the vegetables, or during the agriculture, in order to enhance the productivity, we are using the agrochemicals. Now, these agrochemicals has entered into our food chain. They have entered into the entire food web, and it has become an environmental burden. How? Because these, they enter into every step, they enter into every system, which is causing a problem to the organism, which is not only the living organisms, which include human beings, but also other animals as well as the birds, which are affected by us. And if we go back to the 6th or 7th class, we might have studied the food chain. And in the food chain, what happens that there are the different trophic levels. There is the lowermost trophic level where there is someone who produces the food, and then there are the different type of the eaters which are above the chain. Now what happens, whatever the energy is being drawn at every level, it is magnified at the next level, but there is certain kind of the changes happening. Now in case of the agrochemicals, what is happening that their residue is left inside the plants, and this, when these plants they are taken up by the next eaters, what happens that they magnify at every level, and at every trophic level there is the increase of the agrochemical. इस चीज को हम agrochemical poisoning भी बोलते हैं. Now why we call it as the agrochemical poisoning? Because अगर agrochemical root के through प्लांट में जा रहा है, तो वो प्लांट के डिफरेंट पार्ट्स में, चाहे वो हैवी मेटल के थ्रू है, हर्बिसाइड है, पेस्टिसाइड्स हैं, वो प्लांट के बीच में अकमुलेट हो जाते हैं. जब हम किसी प्लांट के पार्ट को खाते हैं, तो उस प्लांट के पार्ट को जो भी ह्यूमन बींग्स कंज्यूम करते हैं, कुकिंग से भी पेस्टिसाइड्स या हैवी मेटल्स उसमें खत्म नहीं होते हैं. सो ड्यू टू दिस, at every level, these chemicals, when they enter into the system, they basically cause a environmental problem, and they are accumulated into the environment. Now what happens, to overcome the use of these agrochemicals, हमारे पास कुछ alternative होना चाहिए. क्योंकि हम लोग agrochemical कहाँ दे रहे हैं, हम लोग दे रहे हैं agrochemicals when we are giving the a different type of the fertilizers, उसमें भी agrochemicals हैं, because they are chemically in nature, synthesized हैं. जब हम लोग जो है पेस्टिसाइड्स का यूज कर रहे हैं, देयर आर आल्सो द एग्रोकेमिकल्स. व्हेन वी आर spraying certain kind of the food crop enhancing products, there are also the chemically based things which are more or less harmful to the living organisms. For this purpose, what happens that we need to find an alternative, and alternative comes in the form of the PGPRs. Because PGPRs की खासियत क्या है कि वो एक ऑलरेडी प्लांट के साथ एक एसोसिएटेड रिलेशनशिप में रहते हैं. कहाँ रहते हैं? दे स्टे इनसाइड द रूट. नाउ इन द रूट्स वी आर हैविंग द PGPRs, which are basically the plant growth promoting rhizobacteria, and they surround the root system. थोड़ा सा रूट के आसपास सराउंड करते हैं, जिसको कहा जाता है rhizosphere. And in the rhizosphere, these rhizobacteria, they occur, they live there, they actively adapt, and they form a very dense biofilms over the root surface, where they basically affect the host signaling, they affect the biochemistry of the plants. Now, how does these PGPRs they work? PGPRs का जो पहला process है, they help into the nitrogen fixation. If they help in the biological nitrogen fixation, we will have the less use of the nitrogen based fertilizers. Then they also help in the solubilization of the phosphate, because plants cannot take that direct nitrogen as well as phosphate. When phosphate is solubilized, plant they can access the orthophosphates, as well as they also help in the release of certain kind of the plant based hormones, which are called as the phytohormones, or which we call as the plant growth regulators, including auxins, gibberellins, cytokinins, which induce the rapid root elongation or the lateral branching of the roots, which increase the root surface area in order to grab more of the nutrition from the soil. Now, the most important area where rhizosphere is the most important area of the root, where there is the microbial hub. Now, what is rhizosphere? Rhizosphere is the area or the zone where different type of the biological as well as chemical attributes are present, and they influence the root secretions. And this rhizosphere is also the hotspot for different type of interactions of the microorganisms and the soil. Now, in the rhizosphere, different type of the microbial groups are available, which are the bacteria, fungi, protozoa, algae, which affect the activities around the rhizosphere. And when we are talking about only the bacteria which are inhabiting inside the rhizosphere or the rhizobacteria, these are called as the plant growth promoting rhizobacteria or the PGPRs. Now, if we see at this particular diagram, here we can see that this is the root. Near the root, I have drawn a different type of the bacteria. Now, what happens that if we see the root structure, the root is having the root here. Then, corresponding to these root hairs, a nearby zone which is around 1 mm in the area is called as the rhizosphere. Now, rhizosphere can be the endorhizosphere, which is inside the root, exorhizosphere, which is nearby to the root, and then there is the root tip, and the area which is beyond the activity of these microbes, it is the non-rhizosphere soil. Now, near this rhizosphere soil, the activity of the microbes is the maximum. When we talk about the microbes which are near the plant root, they are called as the PGPMs, that is the plant growth promoting microbes or the microbial flora. And we can also see another term which is the PGPB. The PGPB is plant growth promoting bacteria, but the most commonly used term is the PGPRs. So PGPRs are basically the plant growth promoting rhizobacteria, which occur in a particular relationship in the root rhizosphere or the root area where there is a particular different type of the environment exists. Now, how does this different type of the environment exist? Because roots, they secrete the root exudates. Now, when the roots of the plants, they secrete the root exudates, they have certain kind of the biochemicals, which include the carbohydrates or the sugars. They have amino acids. They do consist of the organic acids. There are secondary metabolites like flavonoids and also proteins and the fatty acids, which are more in the root exudates. Now, these root exudates, they are important. Root exudates means secretion of the roots. They are basically very low molecular weight compounds which are non-metabolically released. Now, this non-metabolically released is important because they are not the product of the direct metabolism, primary metabolism. They are released non-metabolically through the roots. Now, these exudates, they are again very crucial because they act as the signals. They act as a signal because they are responsible for repelling the microbial pathogens. When they repel the microbial pathogens, they are important for the plants. Why they're important for the plants? Because they basically protect the plants from various attacks of the pathogens. They also act as the rhizospheric messenger. अब messenger किसके बीच में ये role play करते हैं? They play a role between the plant root and the rhizobacterial organisms. Now, there is a specific growth which is triggered by these PGPRs, which is being harvested or which is being harnessed by using the PGPRs for the plant growth development. Now, if we see in the rhizosphere, not only root exudates are there are also some metabolically released compounds. Root exudates are non-metabolically released compounds, whereas the other compounds, which are the metabolically released compounds. They include the lysates. These lysates are released from the cells during their autolysis, or the mucilage is also released in the rhizospheric zone. Now, when these secretions, which are metabolically released secretions, they act as chemoattractants, and they attract the different type of the microflora. And because these metabolically released compounds, they attract the microflora, due to which PGPRs, they enter the root. When they enter the root, then inside the root, they set up the endophytic association. And due to this endophytic association, they overcome the barrier which is occurring due to the endodermis. Root endodermis causes the barrier for the inflow of different type of the microorganisms. But due to the endophytic association, these PGPRs, they overcome the endodermis barrier and they enter the root, by which they further moves to the above ground parts, to the leaves, stems, or the other organs. And then due to these endophytic relationships, we can know about the different type of the PGPR, which can be used for a particular crop to enhance its productivity. Is it clear? Can we move further? That is the next part, or the part of my presentation, that is PGPR for the sustainable agriculture. So sustainable agriculture is kind of the green agriculture, I must say. Now, the green agriculture is required. Why it is required? Because the population of the world, it is increasing, and we are mostly dependent upon the agrochemicals. And if we want to reduce the dependency on these dangerous agrochemicals, we need to harness the PGPRs. And when we need to harness these PGPRs, we need to focus upon some alternatives like sustainable agriculture, where we can have the farming system, where a kind of a organization, an agriculture where we can have the food for the need of the population. We have the fiber or other resources for the need of the generation, and it will not compromise the ability to generate the food and fiber for the coming generations. At the same time, environmental health, economic health, and the profitability, social equity shall be maintained. And for this ecosystem resilience or the climate resilience, the sustainable agriculture is being a topic which is being thought over by the different kind of the researchers. And as a product of this, we are looking forward for the different methods by which we can change the structure of the soil. Now, why we need to change the structure of the soil? We need to restore the soil health. And this soil health can be restored by engineering the rhizosphere, by changing the structure of the rhizosphere. And we can change the structure of rhizosphere by introducing the microbes, which are the PGPRs, which produce extracellular polymeric substances, which can attach together, and by getting glued together, they create highly resistant macro aggregates inside the soil. And these aggregates, they optimize the soil aeration, they increase the water retention capacity, as well as they reduce the soil erosion vulnerability. And as a result of the different studies, it has been observed that when we are using the PGPR with 50% of chemical fertilizers, we are having around 95% of the target yield which we are expecting for the particular crop. When we are using alone agrochemical or the chemical fertilizers, it is hardly 90% of the target yield. And some of the studies, they have also revealed that the PGPRs alone, without any chemical input, they are giving us around 78% of the productivity of a particular crop. and it basically implies that PGPRs can be used with the chemical fertilizers to enhance the productivity, and they can be used alone, and some changes at the PGPRs or to know the genera of the PGPR is important. And if we focus, there are some genera of the bacteria which I'm discussing here, in which 4 major genera are there. Bacillus species is there, Pseudomonas is there, Azotobacter is there, Rhizobium is there. Now, Bacillus is used for the wheat, maize, and the potato. It has the major benefit because it helps in the phosphate solubilization, it helps in the endospore formation, and it is very highly advantageous for the agriculture productivity, and it enhances the shelf life sustainability of the crops as well. The Pseudomonas has the quality that it produces the siderophores, it induces the systematic resistance, and it has been used in the rice, soybean, and other vegetables, where it has highly increased the rapid root zone colonization, and produce of this crop has been increased. When we have used the Azotobacter in the sorghum, millets, and the cotton, it has increased the organic carbon in the soil. And Rhizobium, when it has been used in the chickpeas, chickpea is a legume crop. When it has been used in the chickpea, now in the legume crops already there is the rhizobacteria, and when Rhizobium is again given to the chickpea, it has very highly increased the nitrogen source in the chickpea. So we have the different type of the bacterial genera of the PGPRs which can be used. Now, when we talk about these PGPRs, they basically help. Why and how? Because they reduce the impact of the bioaccumulation of agrochemicals, they reduce the impact of the ecosystem deterioration by the agrochemicals, they reduce the impact of human health risk which is imposed by the agrochemical, as well as they have been found to decrease the impact of the commonly used agrochemicals which release the heavy metal and the pesticide. We have seen and we have collected the different type of the agrochemicals. When they have checked, it has been found that in number of the agrochemicals, there is an increased amount of the heavy metals, and in the pesticides, they have their own problem because pesticide already gets accumulated into the system and they cause changes in the plants. So this is one of the impact which we have drawn and has been sent to one of the paper. But we have found that these heavy metals in the plant root, they get mobilized, they get absorbed on the root surface, due to the absorption on the root surface are sequestered in the root cell vacuoles, and through the xylem loading, these heavy metals enters and they go on the above ground part of the plant shoots. And when they reach on the above ground, above level parts of the plant shoots, they basically gets accumulated and they deteriorate health, and they cause the oxidative burst, osmotic alterations, and reduced photosynthesis in the affected plants. Now, when we were studying about the impact of these heavy metals and the pesticides, we have seen that we can use the PGPRs. And these PGPRs, what they do, they tolerate and the heavy metals. They enhance in the bioremediation and the detoxification of these particular toxicants. They reduce the oxidative damage. And when we are using more than 1, 2 or more strains, they again change the productivity or the growth of particular plant. And we can formulate the different types of the PGPRs which can be developed be used for a particular crop, which can and when we study the different crops at the different environments, a particular formulation of the PGPR can be used to enhance its growth, development, and the productivity. So if we look at this particular slide, what we have done, these red HM stand
हेलो मैम, आपका कनेक्शन एरर हो रहा है, चेक कर लो एक बार।
in the soil and we release the particular type of the formulation of the PGPRs, what happened that heavy metals, their accumulation was reduced, as well as the pesticides, they were changed or they were modified. Now, there are the different type of the combinations of the PGPRs, because if we are using the PGPRs in combinations, they help in the productivity enhancement of particular crop. Now, these PGPRs, they can be used with the mycorrhiza. Already, a few researchers, they have worked upon this. And then these PGPRs, they can be used along with the biochar, and successfully they have been used with the nano agents. And PGPRs can be used with the earth forms like Escherichia phytid. Are showing the root exudates. Now what happens that in the roots or near the roots, there are the pesticides. Now, when there is the maximum amount of pesticides, which is nearby the root, and if there are more number of the rhizobacteria in the roots, these rhizobacteria, they can help in the pesticide degradation. And by degrading the pesticides, they make them less toxic pesticides or the less toxic intermediates. And these rhizobacteria, they can also convert the pesticide by mineralization into dioxide or the water. And in this, the harmful pesticides, they are converted or the biodegraded by the PGPRs or the rhizobacteria. And when it atmosphere or it causes the bioremediation near the root system, and the plants, they absorb the nutrients which are having the less amount of the pesticides. Now, again, another thing, this is in case of the heavy metals. How does these PGPRs, they act during the heavy metals or the pesticide stress? In the heavy metals, these PGPRs, they restore the levels of reactive oxygen species. When they balance the amount of reactive oxygen species and the antioxidant enzymes, it helps in the recovery of the oxidative burst. Another thing, what they do, they improve the photosynthetic attributes or the chlorophyll contents, etc. in the plants. They immobilize the heavy metals by reducing their translocation by various methods, which can be the complex or the chelate formation or the adsorption or precipitation. By this, these heavy metals, they are not translocated to the other parts, and in return, when all these things happen, they help in the enhancement of the plant growth. We have already discussed that how does they degrade the pesticides near the roots, and they help in the plant growth and development. This is another study. When we were working on the chickpea, we have observed that during the salt conditions, the plants, they have very reduced growth. Now, when we have given the PGPR during the salt conditions, why salt conditions? Because the soils of agriculture areas, they have been found to have the rich salt. They are saline. Saline due to the use of the agrochemicals, saline due to the use of certain kind of the pesticide, etc., as well as the due to the leachates or due to some other factors, the maximum areas of the India, they have the saline soils, and this salinity, it causes the problem for the growth and the development of the plants. So when we have seen that due to the salt, the plants, they were less green, they were not very healthy. And when we have given the PGPR, the growth of particular plant with a single PGPR was found to be changed. But when we have given the combination of PGPRs, the growth was changed more. And when we have given the PGPR with microalgae, it was abundant change in the growth. Mycorrhiza addition to the PGPR also increased the growth. And when we have given the earthworm with the PGPR, the growth was also tremendously changed. So we can use the different combination. It depends from a crop to crop. This is for the case of the chickpea. We have found that the PGPR and microalgae, as well as the use of PGPR and the earthworm, has been helpful in increasing the growth, development, and the productivity of the crop under salt stress condition.
Dear participants, ma'am have some network issue, so please wait for a while. Ma'am will rejoin.
Is it visible?
Yes,
Please
ma'am.
is it visible? So
Yes, sir.
can you let me know where the connection went off?
Yes,
Have we done this?
ma'am.
ये हो गया था हमारा?
यस मैम।
ओके, सो कनेक्शन कहाँ जैसे लूज हुआ था, इसके बाद हमने ये नेक्स्ट कर लिया था? बिकॉज मुझे लगता है मेरी साइड से
मैम ये नहीं कर पाए
कनेक्शन
थे।
ये नहीं कर पाए थे। ओके, आई थिंक वी हैव डन
ये
दिस
नहीं
वन।
था।
हम हैवी मेटल्स तक एक्सप्लेन कर दिया था आपने।
हैवी मेटल्स हमने कर दिया था। वी हैव डन
जी
दिस
मैम।
थिंग आई थिंक। सो वी हैव डन द बायोडिग्रेडेशन ऑफ पेस्टिसाइड्स।
23
सो वी हैव
24
डन दिस
स्लाइड्स।
वन। हाँ जी, ये हो गया था, ये भी हो गया था।
जी मैम।
एंड वी हैव डन दिस वन।
ये
नो,
स्लाइड विजिबल नहीं हो रही मैम।
ये स्लाइड विजिबल है?
नहीं मैम।
ये विजिबल है, ये नहीं हो रहा है। दिस वन इज विजिबल।
नो
अभी
मैम,
विजिबल है?
स्लाइड नहीं हो रही। नहीं मैम।
मैं ऐसे ही बोल दूं क्योंकि जैसे मैं स्लाइड शेयर कर रही हूँ, बट
कोई मैम ऐसे एक्सप्लेन कर दीजिए आप।
okay. So basically the next slide which I have drawn, that is for the combination of PGPR with, let me try once again because that is very important result of one of our study. अगर हो जाता है तो ठीक है, otherwise I'll. Now is it visible?
यस मैम, विजिबल है अब।
Okay.
Yes,
So-
ma'am. It is visible now.
Okay, so this is basically the result of one of our study on the chickpea. Chickpea पे हमने काम किया at a salt stress, and what we have done, we have used the different blends of the other micro or the macroorganisms with the PGPR. We have given the salt stress. Now, why salt stress was chosen? Because maximum soils of India, they are affected by the high salinity. And due to the high salinity, what happens that the productivity of the crop or the growth of particular crop is being reduced. Now, first one is the diagram which we have drawn for the plant which were under the salt stress or
Sorry to
under the stress.
interrupt मैम। मैम अगेन द स्लाइड इज नॉट विजिबल। मैम आप ऐसे ही एक्सप्लेन कर दीजिए।
Okay, so what happens that when we are basically giving a particular stress to the plants, under the stress conditions, under the stress conditions, the crop is being affected, and when we are giving the PGPR, they have enhanced the productivity of the crop. And when particular PGPR was given, I think again it is not visible, so I'm leaving the slides. We are just going to discuss and it was prone to the pathogens, and then there was the less osmolyte accumulation as well as the low adaptability. Now, during the salt stress conditions, the growth was affected. But when we have given the PGPRs, which were given as the rhizospheric fungi and the rhizospheric bacteria, the growth and the productivity of the plant was increased, oxidative stress was found to be reduced, and nutrient status of the crop was enhanced, and there was the better stress adaptability for a particular plants, and there was the accumulation of the osmolytes, proline was also accumulated, and growth of the plant was supposed and found to be increased. Similarly, we studied certain kind of the halo-tolerant plant दोबारा से जो है वो आपका ज्वाइन हो गया, ठीक है. सो इफ इट इज बीइंग ज्वाइन, तो आई थिंक मैं बिना प्रेजेंटेशन के ही इसको कंक्लूड कर दूं.
जी मैम, कर दीजिए.
कैमरा ऑफ करके तो शेयर कर सकते. ओके, एक बार मैं कोशिश करके देखती हूँ. आई हैव चेंज्ड माय लोकेशन क्योंकि मेरे घर पे अभी तो नेट चल रहा है. देयर इज ओनली सम प्रॉब्लम हियर. लेट अस सी. आई विश दैट आई कैन कंप्लीट इट. प्रेजेंट हो रहे हैं. ओके ओके, सो आई एम डूइंग दिस वन. Can I take only 10 minutes? Okay, thank you. Okay, I'm extremely sorry. There is some network issue which is happening in my area, but it is only at my desk where I'm working. So I'm extremely sorry for this inconvenience. If I'm visible right now?
No ma'am.
I'm not visible. My presentation is not visible to you, but you can listen to me.
Okay ma'am.
Let us try once again. I wish that we can do it, but I don't know where the problem is occurring. So is it visible now?
Visible now, yes ma'am.
मैं इसका स्लाइड शो नहीं ऑन कर रही हूँ. मुझे कहीं लग रहा है कहीं स्लाइड शो से तो प्रॉब्लम हो रहा है. So let us see. This is the slide which we have discussed, that under the salt stress in the chickpea, but we found that when we given the PGPR along with the microalgae, with the mycorrhiza and the earthworm, the growth was increased even under the stress condition. So it means we can use the PGPRs along with other organisms, which can be the micro or the macroorganisms. They can help in the enhancement of the growth during the stressed conditions, and thus they can be used as the alternative for the plant stress protection. This is another case study when we used the Brassicaceae family, and in the Brassicaceae family, we have seen that under the saline conditions, there was the lesser growth and the yield of particular Brassicaceae plants. ROS was accumulated. There was increase in the oxidative burst, reduced nutrient uptake was there, and these plants, they were affected more by the pathogens, and osmolyte was very lowly accumulated or least accumulated, and these plants were less adaptable. And now what happened when we gave the rhizospheric fungi and the bacteria during the salt stress condition, growth and the productivity of the plant was increased because the ROS system accumulation and their squenching was maintained, and there was the reduced oxidative stress. Nutrient status was enhanced, stress adaptability was also enhanced, and there was the accumulation of osmolytes as well as improved redox status, and it implied that rhizospheric fungi as well as the bacteria, they can be used in the Brassicaceae family. And besides this, we have certain kind of the halo-tolerant plant growth promoting rhizobacteria. Now, these halo-tolerant means stress-tolerant PGPRs, they are important because they activate the genes under the soil stress conditions by changing certain kind of the defense related activities. ROS detoxification they do due to the ROS detoxification genes. They activate the salt stress marker genes. Also, they activate the iron homeostasis related genes and plant growth hormone related genes. It then impact the plant hormones, and they change the exopolysaccharides, volatile organic compounds are deposited, osmoprotectants they are deposited, and antioxidant enzymes they are also deposited. Proteins as well as the ACC deaminase activity, they have been enhanced by the halo-tolerant PGPRs. And this is again, it has been observed. This I have taken from some another paper for which I've given the link. I've already shared this presentation with Mr. Ashutosh. You can take these particular slides from him for yourself. And in this case, we can see that PGPRs, when we are giving, they basically affect various type of the hormones like cytokinin, indole acetic acid, as well as they change the antioxidant levels, they affect the activity of ACC deaminase, they change different levels of ABA, ROS, ethylene, etc. during the salt conditions as well as the drought conditions, and they change the fertility of the crops by changing the root morphology, transportation, and elevating the phosphate and nitrate accumulation. So apart from the salt, pesticide, heavy metals, during the drought stress also we can see that
Dear participants, due to an unexpected internet connectivity issue, today's sessions by Dr. Indu Sharma on PGPR for sustainable agriculture could not be completed. We sincerely apologize for all inconvenience caused. To ensure that all participants receive the complete learning experience, the remaining part of today's sessions will be rescheduled and conducted on another day. The revised date and time will be communicated well in advance through our official WhatsApp group and email. We appreciate and patience, understanding, and continued support. Thank you for being a part of 21-day International Summer School Training Program on Climate Resilient Agriculture and Allied Sciences. You may leave now.